Hello all, welcome back to the BioRock channel. In this video today we are going to learn about the translocation of sugars. So it's all about the translocation of sugars in plants, okay, through the phloem. And the principle that we learn here, the theory is going to be the mass flow hypothesis. Mass flow hypothesis. So we are going to learn about this. So before I explain you the mass flow hypothesis, children, uh, I'll give you this in a very easy method to understand what is actually happening. Okay, so let me take this particular side as a source. Source is nothing but where the sugars are manufactured, okay, by the photosynthesis process. So obviously that is going to be our leaf, and we'll take another end which is going to be a sink. Sink is the one which is going to utilize the sugar synthesized and transported by the sources. So they are using up, these are using up the sucroses, using up the sucroses for their cellular respiration process. They might be growing shoots, okay, growing shoot parts, or it might be a root. Okay, so if it would be a root will be using the sucroses for the future use. So they will be storing the sugars, okay, and these stored sugars are going to be actually in a insoluble form. They are going to be in a insoluble form. So either you can the zinc can be a root, okay, or it can be any other growing parts, okay, it can be any other growing parts of the plant, parts of the plant. Actually the movement of the sap is not only in one direction, okay, it's dual direction. It can happen based on the conditions. But now what we are trying to learn is only a uniflow. It's going to be the phloem is going to be the one which is going to transport these sucroses towards the sink side. So this is going to be my phloem. And the leaf, what I was talking about as a source, is synthesizing glucoses by the photosynthesis process. But since these glucoses are highly reactive, they have to be converted into a least reactive or less reactive form. And that less reactive form is going to be the sucrose. Okay, so the sucrose. So we are going to consider about the sucroses here. So since continuously they have been produced here, the sucrose levels are very high, while the water concentration towards the source side will be less, because of which we can consider that the water potential here is also going to be less. Isn't it, children? Now, towards the sink side, the sucroses are actively being used for the cellular respiration process. So the sucrose levels are going to be less, whereas the water concentration will be more. Therefore, obviously, the water potential is also going to be more. This is the condition here. Now, what is this is, these are the adjacent cells which are being present in the plant. And let me even take, we all know that besides the phloem, we are going to have even the xylem which is present. So this will be my xylem which is being present, children. And the others are the cells which are being present. All the adjacent, all this adjacent portion is going to be the cells of the plant body. Now, since the water potential is less here, the adjacent cells are going to have a water potential more. So due to which, that is due to osmosis, now the water molecules will take entry into the source. So due to which what happens now? Due to which the water concentration slowly increases. Okay, so the water potential from higher water potential to lower water potential, the water molecules are moving. So from the surrounding cells, the water is moving into source, thereby increasing the volume of water. If the volume of water increases, hydrostatic pressure increases. Hydrostatic pressure is going to increase, and this is that hydrostatic pressure which is going to drive the sugars and water from the source side towards the sink side. This is mass flow hypothesis. Okay, children. Now, what is happening here? The hydrostatic pressure is high, so it's pushing the sap. But where does it push? It pushes towards where the hydrostatic pressure is less. That is going to be your sink. How is that? See now, the water potential inside is high, but the water potential comparatively outside is going to be less. So obviously the water from higher water concentration moves into lower water concentration. So the water molecules are moving out. Thereby what happens now inside the sink, now the water potential drops. Thereby the volume of water is also going to drop. Thereby the hydrostatic pressure in the sink side decreases. So obviously now the things move from a higher hydrostatic pressure side to a lower hydrostatic pressure, that is going to be sink side, and this is going to be your source side. This is going to be the funda for the mass flow hypothesis, children. Now coming on to explaining this procedure in detail now, let me take xylem as well as phloem also. Let me draw this for you all. Then I can explain this procedure well to you all. So this is going to be my xylem. Xylem is going to have only water molecules. So it obviously is going to have a higher water potential. Okay. The next thing that I'll consider is going to be the adjacent vascular bundle, which is going to be our phloem. So this is going to be my phloem, children. And the phloem is made up of your sieve elements. Okay. And the sieve elements are going to have the sieve ends, which are going to help in movement of your sap. Okay. And besides these, you are going to have their own other cells called as companion cells. So let me take this as a companion cells. Companion cells and our sieve elements are connected by plasmodesmata. Okay. And next, let me take your source, that is a leaf. Okay. This is going to be my source. We consider the leaf. Now here also in the sink side, we are going to have again the companion cell, because your sieve elements are been associated with the companion cells here. The companion cells. And what I have is going to be a sink. Going to be a sink. So this is going to be your sink. Now children, in the source, that is your leaf here, we are going to see some special transport proteins. Okay. So let me draw. So this is your transport protein, children. Okay. What is this? This is a, how do I, yeah, this is going to be your transport protein. Okay. And we all know that our sources are continuously synthesizing sugars. Sugars. Remember that glucose, sucrose, amino acids, all are large molecules. Large. Large sized molecules can only be transported to transport proteins. They can't be transported directly through the membrane. Okay, they need a transport protein. And what will this procedure be called as? If something of large size is being transported through the transport protein, we call this as facilitated diffusion. Facilitated diffusion. So from a higher concentration of sugars, now the sucrose is being moved out of the source cell through a transport protein. This is my transport protein. So it's been diffused out into the lower concentration of the companion cells. But how is that happening? Let me show you now. Now what I'll take is, I'll take up co-transport. Companion cells are going to have co-transporters. They are going to have co-transporters. Co-transporters are those which are going to transport two things at once inside towards one direction. Okay. And remember, companion cells are going to have many mitochondria also. Companion cells are going to have many mitochondria. They're actually means they're producing ATP for themselves, which is going to facilitate what we are going to call as a active transportation, children. Now in the companion cells, what we have is proton pumps. This is a proton pump which is going to pump the proton out into the extracellular space here. This is extracellular space. Extracellular space. Now what happens is sucroses are going to be moved into, as well as protons are also going to be moved into the companion cell, and this is being done by whom? By a co-transporter. Okay, so companion cells are going to have co-transport proteins. Co-transport proteins which are transporting whom? The sugars as well as protons from out to inside. Okay, at once. And these proton pumps, actually what do they use is, they are going to use ATP to pump a proton out into the extracellular matrix, children. Therefore, translocation is a active process. Translocation is active process because it is utilizing ATP. Did you understand? Companion cells are having mitochondria, they're synthesizing ATP, and this particular ATP is used to move the protons from the companion cells into the extracellular space, from where they are co-transported with the sucrose molecule into the companion cells, children. Now what is happening is the sucroses are getting moved from the source into the companion cells. Now slowly the companion cells are going to be diffused into the sieve elements by a process of diffusion. So this procedure is going to be the diffusion process through plasmodesmata, children. So what is this here is going to be plasmodesmata. So this is going to be your plasmodesmata. Plasmodesmata. Even here, the sieve elements are connected with the plasmodesmata. Okay, so this is important. So slowly now the sucrose are being moved into the what we call as let me color this well such that it will be easy for you to remember that this is a plasmodesmata. Okay, that's a plasmodesmata through which your sucroses are moved. As the sucroses are getting moved here, the sucrose concentration will increase and the water concentration will decrease and the water potential will decrease. And here the water potential is more. So because of the osmosis process now, because of the osmosis process now, water will move from higher water potential into lower water potential area. So now the water volume increases, thereby increasing the hydrostatic pressure inside the sieve elements to be very large. Now, because of the more hydrostatic pressure here, the sap is going to be moved further down through all the sieve elements, okay? Through all the sieve elements. So now the sugars and the water moves down because of the higher hydrostatic pressure. And all the sucroses which come over here, now what they do is they are actively transported into the sink here, children. The sucroses are being moved actively, they are being actively transported into where? Into the sink side. So as the sugar levels will increase here, the water concentration will decrease, thereby the water potential will decrease. And obviously, since sugars are moving here, the sugar concentration will decrease and the water potential will be high. So again, which process will occur, children, now? It is going to be which procedure? It's going to be osmosis. So obviously, because of the osmosis process, the water is making movement towards this particular sink side. From where? From here, from your sieve element, and little amounts of the water even moves into the xylem because continuously the water is getting transpired out into the atmosphere. So because of osmosis, little amounts of water even enters into the xylem, and the water even takes entry where into your sink side along with the sucroses. So this is a procedure of the mass flow hypothesis. I hope, children, you have understood how the transport proteins, co-transport, you remember the co-transport proteins I have introduced you when I was teaching you the excretion and osmoregulation and the reabsorption process. I have introduced you with the co-transport proteins, okay? So this is a one that you need to understand and remember. Now what I'll do is I'll try to share my screen with you all, children, so that I can show you a slide which will give you the idea regarding the translocation process. Okay, so this is the one what I need to introduce you, children. So you can see the source, the leaf having the chloroplast, synthesizing the sucroses. They are transported into the, what you call its companion cells through the co-transporters along with the proton pumps, back to transpiration of proton. So proton and the sucrose together into the companion cell, but the sucrose gets out of the source through the facilitated diffusion and then into the phloem. And because of the high water potential in the xylem, the water enters inside and hydrostatic pressure increases, which moves the sap further down towards the sink side. It moves actively transported inside, and the water potential reduces here. Obviously, it moves from a high water potential to the lower water potential, and it even takes entry into the xylem vessels also. So this is the translocation of sap, what we call as mass flow hypothesis. I hope, children, you all have understood this procedure. If you have liked the video, please do hit the like button. Try to share my channel with others and subscribe my channel. And do please comment in the comment section how you have liked the video. Thank you all. Thank you.